Great. I'm really looking forward to talking to you guys today. Um, if you guys can give me screen sharing and we will jump right in. And I'll ask you guys to be slightly patient with me today. I got my COVID vaccine second Pfizer shot yesterday, so I'm mostly fine, but I have a little bit slower than usual today.
No worries
So
at all.
I will use that as my excuse for how we're doing this today. But welcome everybody. I want to give you guys an overview, introduce myself, and give an overview to these topics to make you really think about them. I like to make these interactive sessions, so I will be opening up the floor for questions throughout the talk. I'll let you know when I want to have some discussion, because to me, cell and gene therapy is something I've been involved with for a long time, and I'm really excited to present this kind of general overview from my viewpoint, which is not the viewpoint of a scientist. So, who am I? Why am I giving a talk about this if I'm not a scientist? My name's Maria. I am the president of BioCurious, which is one of the world's oldest community biology labs, or like a makerspace for biotech. It opened in 2011. We've got a 7,000 square foot research space, which includes tissue culture lab, on-site gene sequencing, on-site HPLC protein purification, and more. So we're actually better outfitted than most of the incubator spaces in the Bay Area. We're located in Santa Clara, California, which is where I am. I'm also a board member of the Momental Foundation. We're a nonprofit foundation that pairs grad students with startup companies. So we take STEM grad students who are postdocs and PhDs, and we pair them with startup companies who need some research help. I also co-lead a lot of different organizations. Oh, you know what? Let me put this in presenter mode and you guys can actually see this a little bit better. There we go. That looks better. I co-lead the Global Community BioSummit as one of the meta leads. It is a large global conference for community labs around the world. Our 2021 conference had 650 attendees from 62 countries. As part of that program, we run a leadership fellowship for teaching people leadership skills in science and how to start their own labs. We're on our third year of that leadership fellowship. I also co-founded, I'm a board member of Real Vegan Cheese, a project to make recombinant vegan cheese and yeast. I am a member of the Open Insulin Foundation, which was started just over 5 years ago, where we're trying to make open source insulin and bring low-cost medicine to people in a decentralized model. I also lead a lot of community projects. These are projects that are open to anyone of any age, and the longest of them has been running 10 years. So DIY bioprinting. We also do Kombucha Genomics, open source lab hardware, Cuttlefish Research. I do a lot of public speaking about biosecurity and bioethics. However, as I mentioned at the beginning, I do not have a background in science. My bachelor's degree is in operating and programming. And I do have an MBA in global management. I spent about 10 years working in Silicon Valley in tech, companies like Apple, et cetera, and kind of wandered my way into biotech. I am a full-time stay-at-home mom, which is how I can do this many things. I have a 13-year-old, a 16-year-old, and I have a lot of hobbies on top of all the other things I do. So all this to say, I'm not an expert on the science. I have been publicly speaking about stem cell research, gene therapies, et cetera, for over 15 years. And I think it's really fascinating. So let's jump into this today. So cell and gene therapy, these are actually a little confusing because sometimes people don't really understand what they are. They're overlapping fields. The overall aim of these fields, like anything in medicine, is to treat, prevent, cure disease, but they actually approach this from two different methods. So a lot of people ask, aren't cell and gene therapies the same? No. So cell therapy wants to take cells and use them to either carry a therapy throughout your body or cultivate them and inject them into the patient. They can either be the patient's own cells that have been modified and injected back into them, or donor cells. whereas gene therapy wants to treat the disease, often genetic disorders, by inactivating or introducing new genes into your body. There's a lot of different ways to do this. There's a lot of science behind what's happening here. Where it gets really confusing is there are some therapies that are actually both. So they're altering the genes of specific type of cells outside the body, and then you reinsert them into the body. So all of that to say, this can be confusing. I don't know what the general background of the people attending is. I was told you guys would be at least high school biology and up. So what we're talking about here are cells and genes. So, I think this year, over a year into the pandemic, we know a lot about cells. We've been talking about genes. We've been talking about COVID for a long time. Our cells have a nucleus in them. And in that nucleus is our chromosomes. And in those chromosomes, they're made up of DNA, which is made up of genes. So that DNA doesn't always work right. And sometimes there's problems with the cells themselves because of disease, often cancer. And we need to find ways to either fix a single piece of DNA that's wrong, that's causing a problem in a cell, or fix the whole cell. So let's talk about gene therapy to start with, because this goes back a lot further than genetic engineering. So the history of gene therapy, this is not an overall history. We could do that for an hour. I just want to give you guys some highlights. So 1970s, a researcher named Rogers was cited for proposing the idea that exogenous DNA could actually replace defective DNA in those who are suffering from genetic defects. So he was the first person who actually talked about this. So this is not a new concept. In 1972, Friedman and Robin, they authored a paper for Science titled Gene Therapy for Human Genetic Disease. So that was the very first time we actually heard the phrase gene therapy. Fast forward a few years, almost 2 decades. In the 1990s, we had our first approved gene therapy research in the United States. So 4-year-old Ashanti da Silva got some treatment for a genetic defect that left her with ADA-SCID. So this was an immune system deficiency. And what they did here is the defective gene in her blood was replaced with a variant gene that was functional. However, this worked really well, but it wasn't a long-term treatment because what they did is they treated the disease. They didn't actually give her a cure for the underlying problems in her immune system that were causing this. So what this led to is her needing lifetime treatments about every 2 months. But it was successful. It just wasn't the permanent sort of cure that they were looking for. But it was really, really good research. So let's go forward again, another decade. In 2003, Gendicine was a cancer gene therapy that delivers tumor suppressor gene P53 using an engineered adenovirus. So for those of you keeping up with our COVID research, adenoviruses are being used a lot to deliver things into our cells. The Johnson & Johnson, the AstraZeneca, and other variants of the vaccine are actually using deactivated adenoviruses to get that vaccine into your cells. So this is not new technology. We've been using adenoviruses for a long time. Here they wanted to use adenoviruses to actually deal with P53. For those of you who may not have heard of P53, the majority of cancers in humans are some variant of P53 deficiency or problem. So P53 is an oncogene. It's the primary oncogene that causes problems for cancer. So if you ever get into cancer research, you're going to see a lot about P53 or TP53, depending on what you're looking at. So 2006. We had a successful gene therapy to treat 2 adults, patients linked for X-linked chronic granulomatosis disease, which affects myeloid cells and damages the immune system. And this was really significant because it was the first time we actually showed that a gene therapy can actually treat the myeloid system. So this was really, really big. We'd been trying different things. The primary target for gene therapy was and continues to be cancer therapies, but there's a lot of other ways that we can use this technology. 2008, Leber's congenital amaurosis is an inherited blinding disease that is caused by mutations in the RPE65 gene. So 2008, we had a small clinical trial in children. And again, delivery of a recombinant, you'll see adeno-associated virus. So this is another adenovirus way to carry this RPE65 and fix it. So they've been working on trying to use this gene therapy to treat the condition of blindness since before 2008. And I mentioned that specifically because we're going to talk more about some techniques being used today to treat blindness using gene therapies. And 2010. So 2010, we had an 18-year-old male patient in France with beta thalassemia major had been successfully treated. So this is an inherited blood disease where the beta hemoglobin is missing. Patients are very dependent for lifelong blood transfusions. And here they were actually able to do this. Bone marrow transplants are the cure, but the problem with bone marrow transplants is there's not very many people on the transplant list. And so 75% of your patients aren't getting donors. So we need better ways to do this. And this was another way to do this. So they were actually able to modify his own bone marrow cells to create the hemoglobin and cure him of this problem. So a lot going on here. Really of note is 2011 was very, very exciting. It's when the study was published. But in 2007, 2008, a man, Timothy Ray Brown, was cured of HIV. He's often called the Berlin patient. And this was actually done through bone marrow transplant. So he had stem cell transplants, which disabled the CCR5 receptor. We are going to talk about that more later on in the talk, so make sure you're remembering CCR5 receptor. And the fact that he was cured was only accepted in the medical community in 2011. Now, the big thing about this is that he was somebody who was HIV positive. He also needed a bone marrow transplant. They were able to test this idea. They have not been very successful in duplicating the results. The conditions are very, very narrow. And so it has led down a lot of interesting research on this idea of working with the CCR5 receptor. So we're going to talk about that a little bit more. And then in 2015, researchers successfully treated a young boy who had epidermolysis bullosa using skin grafts. So again, this is a pretty interesting technique. So 2015, this is sometimes known as butterfly boy syndrome. They use skin grafts that they made from his own cells and genetically repaired the mutation that actually causes this disease, and were able to fairly successfully treat it. And there was an ongoing clinical trial for this that's happening right now at several sites throughout the world. So very, very brief overview on some of the high points of the history of gene therapies. Currently, as of last year, these are just some of the gene therapies that are available and approved by the FDA in the US right now. So you can see there's not very many, but they do exist. So depends on what you're looking for. Again, if you remember the 1990s, we had that young 4-year-old who was treated for ADA SCID. There is now a standard gene therapy that's available. So that did take just over 25 years to actually bring it to the public. Again, gendosine, which we talked about also from the 90s, is a treatment for head and neck squamous cell carcinomas. So a lot of these treatments have been around. The research on them takes a really long time, but they are starting to finally be available. We're not going to get too far into this, but I did want to make some note on some of the milestones in pluripotent stem cell research, which is separate. So stem cells are not necessarily the only way to do things, but it is running parallel and overlaps a lot with this cell therapy. So in 1961, the first stem cells were described by Dr. James A. Till and Ernest McCullough at University of Toronto. 1996, so many, many decades later, Dolly the sheep was cloned. So that was a big milestone. In 1998, the first human embryonic stem cells were isolated by James Thompson in the United States. In 2006, one of my favorite milestones ever, which is that induced pluripotent stem cells were derived from reprogrammed adult somatic cells using 4 factors. And this work actually led to the 2012 Nobel Prize for Shinya Yamanaka, who is out of Kyoto University and University of San Francisco at the Gladstone Institute, as well as John Gurdon from the Gurdon Institute in Cambridge. And they won their Nobel Prize for figuring out these 4 factors that allow them to take a biopsy of human cells from an adult and actually turn them back into a pluripotent or stem cell state. So they use 4 different transcription factors delivered via virus. And with that, they can take most frequently a skin cell sample, grow up your skin cells once they've got enough cells. They take these 4 transcription factors delivered via virus, and then that actually allows them to create stem cells. This has really, really revolutionized the area of stem cell research. And in the last 10 years, stem cell research has really exploded, especially in California. So California is one of the world centers for stem cell research. But now, that's a lot of science, that's a lot of history. I want to talk about the ethics of all of this science and history, which you might have thought you're beginning to get a lot of science. But cell and gene therapies come with a lot of baggage. So there's a lot of ethics, there's a lot of bioethics concerns, and who has access to these technologies? What are we doing with them? And this is what really interests me about them, is that how does it influence society, and what's happening with them? And also, how do we regulate them? So, we have to start from the basics. How do we define ethics for our science? How do we define ethics for our society? And what do we really mean when we talk about bioethics? So bioethics is sometimes kind of a confusing field to people outside of STEM or outside of medicine, because bioethics includes people from all sorts of fields discussing what's going on, and everything from public policy and law to how do we actually apply this in the clinic. So when we talk about ethics, let's think about what are our big ethical concerns. These are things like, what should I do? How should I act? How should I treat others? What are my obligations or responsibilities towards others? What type of person should I be? Well, when we're thinking about bioethics, are we required to not only not harm others, but are we ethically required to act in a way to make their lives better? What does it mean to treat people fairly? What does it mean to have equitable access to these technologies? How do we design clinical trials that are ethical and better designed? For example, right now there's a big ethical question that is very unresolved in Ohio. Ohio right now is incentivizing vaccines by having a lottery. So they are incentivizing people to get their vaccines, which is a good thing, by giving them the opportunity to have payment for these vaccines. And that's where it gets into an ethical gray area. Incentivizing people who don't have something that you could give them something if they do something with their bodies is kind of an ethical gray area. However, if it's for the good of us all, because we want more people to be vaccinated, again, very, very big gray areas. Some other good questions is, can minors make their own healthcare decisions? How much can they do that? How much input should they get into their healthcare decisions? This is really big bioethical question right now being legislated around the country as far as trans rights. So what are the questions around gene therapies? What is a good use and what is a bad use of gene therapy? And I want you guys to keep this in mind when we're going through this, and please put any questions you have around this into the chat or your thoughts. I'll be taking a look at that in just a few minutes. Who decides which traits are normal and which constitutes a disability or a disorder? So this gets into questions of ableism. This gets into questions of what is a disorder? Who defines what the standard is? Gets into a lot of racial issues as well as, is this normal for some people? Is it not normal? What should we be doing? Is the high cost of gene therapy making it only available to the wealthy? One of the most successful treatments we have right now for cancer is CAR T-cell therapies. CAR T-cell therapies are only available for cost of around $2 million a treatment. No healthcare is covering that cost. So, you know, we've got a great treatment if you are extraordinarily wealthy. Could the widespread use of gene therapy make society less appealing to those who are different? Will this create differences amongst nations? This is something I think about a lot. This is something I discuss at different conferences I go to a lot. Who gets access to these technologies? Who sets the bar? We already see a big problem in society with colorism, with the way that we view who is acceptable and who isn't. As we can design designer babies, what is going to happen going forward? What is going to happen with people who can also use these therapies to create super soldiers, to create super athletes, etc. So what happens if your country, if your people, if your religion doesn't give you access to these sort of therapies? Does that mean that you're less of a person, that you have less rights, etc.? And that gets us down to one of the stickier questions is, should people be allowed to use gene therapy to enhance human traits, things like height, intelligence, athletic ability, and beauty standards? So all of this is on the table. Some of this is possible now, some of this is not. Some of this is coming in the next 5 to 10 years. So there isn't enough public discourse about this. So I really like to get people thinking. So, you know, we're going to come back to this. Actually, you know what? Let's go ahead and take some questions now. Let's see if we have any questions in the chat, or if you guys have any questions about any of these big topics about bioethics before we go on. I've been going on for about 20 minutes, so. What do you guys think about the bioethics of things? Anybody out there have any opinions? I'd love to get some questions in the chat or get some conversation going on what are we going to do with these gene and cell therapies? Who's going to have access to them? You guys are so quiet.
I'll add some flavor.
Yeah.
Hi, I'm Aubrey Greenberg, co-founder of Discovery Labs. We have operations in King of Prussia. We have a CDMO for cell and gene therapy, and worked with Tracy in the past. Anyway, I really have strong opinions as to what we should be using these therapies for.
Yeah.
It should be for cancer, debilitating disease, not for physical or other reasons, gender selection and things of the like. That's my first opinion. And then second, in terms of cost, you know, obviously we should try to advocate for the insurance companies covering the cost of these therapies. And as we're working as a CDMO to lower the cost of producing these therapies, but that doesn't necessarily lower all of the years spent trying to develop them and all the costs associated with that. Maybe we'll reduce the cost to produce a viral vector or manufacture a cell therapy. But, you know, in the pharmaceutical companies that may buy out some of these biotechs ultimately are trying to recoup either the cost that they spent developing the therapy or the cost that they spent acquiring the therapy. And so I think until we get a more streamlined process and platform down, it's going to result in these really exorbitant costs for these therapies that unfortunately are justifiable based on the amount of money invested. So as we streamline the therapies and the costs come down, the overall price to the customer hopefully will come down.
Yeah, but I think that's actually a very good point. Is right now a lot of this also kind of overlaps with this idea of personalized medicine, because a lot of these cannot be deployed, mass deployed. They need to be personalized to people. So when we're thinking about these technologies, who's getting access to them? What are we using this for? What is the cost of them? How much are we investing? These are all really, really good questions. And the development time of this is long. So this is significantly longer than development time for bringing your standard drug to market. So it takes 10 to 15 years, about $1 billion roughly, to bring a drug to market. These gene therapies are taking 20 plus years to bring to market. We're hoping this is going to be speeding up as some of these technologies are proven and a lot more of them are coming out. But the lead time in development has been very long. The investment has not been high enough. So we'll have to see. And we know the public is also a little leery. So there's a certain number of people that feel like editing the human genome, the ethical issues around that, as well as the fundamental misunderstanding of these technologies, is going to be a barrier to acceptance as well. So.
Yeah, I have a thought. Can I add something too here? So I've been thinking a lot about this. And while I don't have like the heavy
introduce yourself as well.
Oh, sorry. Yeah, my name is Maya. Yeah, I appreciate the primer. And I think there's two big things. Like when we talk about ethics, to me, it's like the most interesting part, but really like the big ethical discussions to me are not around like, should we cure diseases using cell and gene therapies, right? Like, no, what? Well, I don't have an issue with that, right? The issue to me, and the more like, provocative ethics come around the gene editing. But then I think within all of this is the ownership over genetic information, which is like very much present in our time. And specifically, like I was just talking to somebody who like really works in this world, and they were talking about personalized genomic medicine as this idea that you would go to the doctor one day with your decoded genome, and there would be discussions about like interactions with both your environment and just different like mutations, or what you're like reading that out, and how to me I'm like, what do people think about that? That's fascinating. I'm like, I never even considered like how I feel about that. Good, bad. Is it okay that your like who has access to that is interesting.
Yeah, and I think there's a few things around that. I don't want to get too far afield, but there are people who are encouraging others, and I discourage this, but there are people out there trying to get people to sell their genome as an NFT, so a non-fungible token. So if you want to like cross the streams of like cryptocurrency and what's going on there and your genome, do not do that, okay? I don't even have the amount of time it will take me to tell you why that is a bad idea, but don't do that. There's questions about if somebody has a gene. Here's a thought question for the audience. If someone has a gene for hyperintelligence, who owns the rights to that gene? Is it the person that has it? Is it the scientist who discovers it? Is the pharmaceutical company who manufactures it? So rights to this are big question. I would also say that if there's a lot of value, and a lot of companies says that there's a lot of value in genetic information, not just ours, but if any organism on the planet, who has the rights to that? Who has the rights of discovery? If there's, a medicine in the Amazon that an indigenous tribe is using, and they tell somebody who's coming through about that, and they're able to figure out what the compound in that is, who has the right to discovery? Who has the right to that? So there's a lot of big questions all across this. Also, when you're getting your genome sequence, think about where it is, who's actually sequencing it. If you're doing 23andMe, a lot of these gene sequencing companies, your genomic information is being sent out of the country. Most of it is being done in China. So China has a lot of your genetic information. And there's a lot of info there. So I think Bob has something he wanted to say. Bob, you want to introduce yourself?
Hi, I'm Bob Castalucci. I'm the founder of Partnership to Prosperity. And I work to try to put educators and employers together to get people jobs. And as I listen to the conversation, what really strikes me is cell and gene therapy is something that, in one sense, is very, I'll say, future and innovation oriented. But I guess in the other, as you laid out through the history, it's really not that new. I don't think it's hard to imagine how it is going to become, that capability is actually going to become commoditized.
Yes.
So what I wonder is what people think about how we might better educate everyone in the population about what it is.
Yeah.
Right. I think the whole warp speed and COVID vaccine thing has been fascinating because almost no one in the general population knows that 50% of the COVID genome was figured out over a decade ago with SARS and MERS.
Yep.
So it was already somewhat common knowledge, right? Sorry, I'm getting up. So it concerns me that whether it's the government, whether it's industry, or whether it's academia, with things like this that you can clearly tell are going to be part of the everyday world, we don't seem to be doing a very good job of helping the whole world know about it, what it is, what the pluses are, what the watchouts are. So I'm really curious to hear people's thoughts about ways to go about that, or maybe you think that I'm just really off the deep end. I'd love to hear that too.
I was going to say, I don't want to get us too far afield, but you're 100% speaking my language. This is stuff that I'm very, very much interested in. How do we prepare our next generation in the bioethics side? You know, I'm working with a group as an advisor on an NIH grant that's looking at how do we get the public more engaged in the discussion of what should the regulation of these therapies for gene editing embryos be? There's a big initiative out of Baylor University about that. So how do we engage the public, and what do we do, and what is the public interested in? How do we get, you know, people of all walks of life, of all religions into this discussion? Because right now, the biggest problem with these technologies is they're being developed, discussed, and decided on by people who are not talking to the general public. There's also a big issue of jobs. How do we make sure, if this is the field of the future, that we don't just have the biotech hubs, which are, you know, the two coasts. We've got huge biotech centers in San Diego, the Bay Area, Boston, New York, wherever. How do we get these technologies deployed and create jobs in small cities? How do we really get people interested in these technologies? How do we create decentralized way to do that? Yes, this is 110% what I spend most of my time thinking and working on. So you've got it. And somehow, some way, here we go. All right, so getting back to the talk, and you guys will see I like to go far afield. That is what I like to do. You know, I do enough of these talks. Just sitting and talking and giving a bunch of PowerPoint slides is not nearly as fun as actually talking with you guys about what's going on. And getting kind of this discussion going. And I think that we get lost in the weeds of the science a lot. You know, it's really exciting. I myself get really excited working with stem cells. I get excited about the science. You know, I'm doing a huge research project on P53 and some of the implications of it in cuttlefish. But the ethics are where the people live. And I think that that's gets lost behind a bit. All right, so getting back into this primer, we're just kind of getting you guys thinking about these types of stem cell therapies, so you guys know what's going on. There's generally 3 types of cell therapies that are going on. Allogenic cell therapies are where the donor is a different person. So this would be where you'd get, for example, bone marrow transplants are allogenic cell therapies. You're getting a transplant of cells from someone else to help your own immune system. Autologous cell therapies are where the cells are actually transplanted and are your own cells. And we don't see this as much, but xenogenic cell therapies, where cells from one species are given to another. Most of the xenogenic cell therapies are from animals that are receiving human cells so that they can be used as model organisms, which is its own bioethical problem in and of itself. What happens, and this is currently a problem right now that people are wrestling with, for example, if you create monkeys that have been modified to have human brains. How many sci-fi movies do we have to have about that before we learn our lessons? Types of cells used in cell therapies. Lots of different types of cells used. We can use human embryonic stem cells, neuronal stem cell therapies. These have been tried since the 90s in Parkinson's disease. Mesenchymal cell therapies, hematopoietic stem cell transplantation, and differentiated or mature cell transplantation. So this is to say that the field is very, very large. So let's talk about some actual uses. This is where it gets interesting. If you are averse to gore, you might not want to watch the next image, but I think that it's a really good example. So we talked a little bit about butterfly boy syndrome, and this is the epidermolysis bullosa gene therapy. So what happens here is the person who's born with this has very fragile skin. There's problems with the collagen in their skin, and it causes their skin to be, quote unquote, as fragile as a butterfly's wing. They get a lot of blistering, and their skin just kind of peels off in huge sheets. They currently, all you can really do is bandage the wounds, manage their pain. It's pretty devastating. They call it butterfly boy syndrome because their skin is as delicate as a butterfly's wings. The lifespan, depending on how severe this is, can be quite short because of infection induced by all these wounds. So what they did, as we mentioned in 2015, is they started a therapy where they would take cells from the patient, they'd grow up sheets of skin for grafts, and then they'd actually use retroviruses to actually fix the gene that's the problem. They've been doing this at Stanford. I know some of the folks working on this. It's really amazing. I've seen the sheets of skin growing. And they then transplant the healed skin that is no longer going to have the problem back onto the patient. This is, you know, as you mentioned, cost, one of the more expensive treatments you can go through. The steps are enormous. This is just not a cheap or easy thing, but the end results can be pretty good. And they're still working on this. It's only about 50% effective. And some of that is just getting the grafts to heal well. So if you don't like gore, please look away. But this is what it looks like. So the top line is the patient's back. The middle line here is the control, which is the patient's right arm. So you can see the baseline of how bad these wounds are is extreme. And then the bottom line is with some of the graphs, as well as the top. So you can see, if you look at baseline, they have almost no skin on their arm. They apply the grafts, and at 6 months, the grafts are still there. At 12 months, the grafts are about 30 or 70% there, and they're at 50% at 2 years out. So it's not a perfect treatment, but this is real world people who are suffering a lot, and it's decent. It's decent results. There's a lot to learn. But I think that that's really, really important, that we're looking further afield for what we can do with these therapies when we understand that there's a single gene that's causing the problems. So let's talk a little bit about gene therapies. For gene therapies, there's two different types going on. Somatic, which is where we are altering the genes of anything that is not a germline cell. So germline cells are your sperm or your egg cells. And currently, there is a moratorium on germline research in the United States and in most countries. We will talk about the CRISPR twins at the end here. And what we're doing here is we're looking at gene therapies for genetic disorders, or where we can actually use gene treatments to help people. Sickle cell disease is one of the first things that we've been targeting, sickle cell, is of course an inherited disorder that leads to abnormal production of hemoglobin. It's called sickle cells because the blood vessels or blood cells, instead of being nice and round, take on kind of a half moon sickle shape. And they're not very good at passing oxygen anymore. There's not really good treatments for this. So because we know the gene that causes this, it would make sense. There's actually a lot of studies about this. I wanted to highlight one to talk about why these are so difficult. So Bluebird Bio is a company that's been working on this. They're using lentiviral vector to create healthy copies of the beta globulin gene. However, and this is how these are very tricky, this study has been halted for about 6 months. They are looking to restart it because they did have a single patient who did develop cancer in the middle of the study. So they needed to be sure that their gene editing didn't cause cancer. And the reason we're worried about that is it's a big gotcha for gene editing. Cancers are problems where genes in your cells are misedited and get out of control. That P53 gene, there's a lot of other oncogenes out there. And they need to make sure that how they're editing is done efficiently and does not potentially cause cancer. So there was a long-term halt on this for about the last 6 months to make sure that it was not. It does not appear that anything related to the gene therapy they got caused the cancer in the patient, so they will be restarting this. But this was a 6-month halt in a trial that's at a phase 1/2. So these studies can take a long time when you are being very methodical and going through them. But they're very promising. This is just one sickle cell study. There's other studies being done right now using other ways to get virus vectors in, including a CRISPR study that's being done with Sangamo Biosciences. So we've talked about these. The worst problem this field has is there's way too many terms. For the last couple of years, maybe 2 or 3 years, people have been trying to use the term genetic surgery. So the first genome surgeons, I've been seeing lots of headlines with this now, and they want to talk about people who want to create these treatments as actually being genetic surgeons for going in there and altering DNA. So if you ever hear that term, what they're talking about is people who are creating these treatments. One really good treatment is one looking at Best disease. This affects people who can go blind early. It's a genetic disorder which affects a single gene in your eye and affects the cones in your eye. UCSF is currently undergoing I think they're entering a phase 2 clinical trial using CRISPR as a way to actually edit the gene that's going on and actually completely correct the condition. So they have a pretty large cohort of people who are interested in this. And they are calling it genome surgeons. So there is a little bit of a rebranding of this. And some of that is to get people more comfortable with it. So this is part of making it a little bit less like genetic modification and more like this is surgery. So I'd love to see what you think about that. So next, we're going to talk a little bit about CRISPR. I don't know how familiar you all are, so I want to give you a little video real quick, and then we'll get back to some discussion. Let me know if you can hear this.
Bacteria and viruses have been fighting since the dawn of life. So-called bacteriophages, or phages, hunt bacteria. In the ocean, phages kill 40% of them every single day. Phages do this by inserting their own genetic code into the bacteria and taking them over to use them as factories. The bacteria try to resist, but fail most of the time because their protection tools are too weak. But sometimes bacteria survive an attack. Only if they do so can they activate their most effective antivirus system. They save a part of the virus DNA in their own genetic code in a DNA archive called CRISPR. Here, it's stored safely until it's needed. When the virus attacks again, the bacterium quickly makes an RNA copy from the DNA archive and arms a secret weapon, a protein called Cas9. The protein now scans the bacterium's insides for signs of the virus invader by comparing every bit of DNA it finds to the sample from the archive. When it finds a 100% perfect match, it activates and cuts out the virus DNA, making it useless, protecting the bacterium against the attack. What's special is that Cas9 is very precise, almost like a DNA surgeon. The revolution began when scientists figured out that the CRISPR system is programmable. You can just give it a copy of DNA you want to modify and put the system into a living cell. If the old techniques of genetic manipulation were like a map, CRISPR is like a GPS system. Aside from being precise, cheap, and easy, CRISPR offers the ability to edit live cells, to switch genes on and off, and target and study particular DNA sequences. It also works for every type of cell, microorganisms, plants, animals, or humans. But despite the revolution CRISPR is for science, it's still just a first-generation tool. More precise tools are already being created and used as we speak.
So that is just a really brief overview on CRISPR. CRISPR is really amazing. Jennifer Doudna and Emile Charpentier won the Nobel Prize for their discovery of CRISPR after some debate whether they should get it or the folks at MIT, Broad Institute. So CRISPR is one way to do genetic engineering. And we don't want to watch that video again. We don't want to watch this one. Come on. There we go. And we hear about it a lot. It is not the first way to do genome editing. It is merely the newest way to do genome editing. However, there has been a big incident with CRISPR, and this is an ethical issue. So I don't know how many in the audience have heard of the CRISPR twins. Let's talk some quick real world ethics that happened in the last couple of years. Chinese researcher He Jinkao and some collaborators unethically edited some embryos in 2017. So at the time, there was a worldwide moratorium, including in the US and China, on gene editing human embryos for implantation. He and his collaborators approached one specific couple with an HIV positive husband and HIV negative wife, along with 6 other couples. He told them that they would be enrolling in some vaccine research. So there's ethical issue number 1. He lied to them about the type of research that he was doing. Ethical issue number 2, the people who were involved had elementary school level education. Did they have the understanding of the science or ethical issues of what they were getting into? He did not undergo any sort of ethical research oversight. He was not admitting to the university, the hospital he was working with, etc., what he was actually doing. They did knock out the CCR5 gene in these twins, so they could try to create twins who would be resistant to HIV. This was not an outstanding issue. This was not a problem. There are ways to actually do in vitro fertilization where the husband has HIV and the wife does not. You can do some simple washing procedures to select for sperm that are not infected with HIV. This is not even that difficult. So he was solving a not big problem. The twins were born in October 2018. He has been jailed for his unethical work in China. However, there is now a Russian researcher with the approval of the Russian government who is planning to pick up a similar experiment and do this in humans later this year or next year. So huge number of ethical issues with the CRISPR twins. They are not the only twins that were born. Some of the other couples have also, it turns out, had twins. This is a wall of text. This is a letter to the editor from some researchers about all the problems with this experiment. So I just want to take a minute to take a look at this. And just talk about it. So we only have a few minutes left, so let's go ahead and get into it. The basic thing here is up, down, and sideways. This was an unethical experiment and children were born as a result of this. Those children, there is no proof that the edits he did would actually protect them from HIV. CCR5 is not the only way that HIV can enter your cells, so it was unethical because it wasn't a good experiment. He did not use the latest version of CRISPR that is better at preventing unintended edits. So that's a second thing. And he didn't do that for cost. He didn't want to pay the full price for a CRISPR cassette. He wanted to use the least expensive cassette he could use. So he was using it like an academia level cassette, not a research quality cassette. He lied up and down all over the place. He was doing this for profit. He was doing this as a company that could then sell this as a service to people. There's just the number of ethical issues about this just go on and on and on and on. But he opened a door, and now other people think that that means that we should start gene editing embryos. So, yeah, there we go. Since we only have about a couple minutes left, let's go ahead and take a couple questions. I'm going to get my power cord because apparently it came loose during the talk. I think my dog knocked it. And see what kind of questions you guys have about gene therapies and what's going on in the world. Word acquired. All right, so anything in the last 5 minutes, we need to wrap up in just a couple minutes. I guess the big thing I just want you guys to take home, and again, sorry, I'm a little scattered today, is that this is a big field. There's a lot of things under this umbrella of gene and cell therapies. The amount of treatments, the amount of research being done is enormous. The number of clinical trials in this area is big. But the number of ethical issues we have is not far away. They are here now. So, I forgot to mention, in addition, for the poor twins, turns out CCR5 may cause problems with their brain development. So that's an additional issue. And it might protect you from HIV, but it does also make you more likely to be susceptible to the flu. So which are you more likely to encounter in everyday life? So I cannot even tell you how much every bioethicist I know has screamed their heads off about this. But it happened, and the twins are out there, and the other twins are out there. And this was not a problem framed in that it was done because it was in China. The problem was that he lied to every institute along the way, including Rice University, where he had collaborators, and Stanford University, who had collaborators. Nobody called him on it until he presented at a conference. So he never thought he did anything wrong, and presented at the World Gene Editing Conference, which happened every 4 years. And 2 days beforehand, a reporter broke the story that he was going to be doing this, and bragging about he was the first person to create human and edited embryos. So he was doing this for fame, for publication, and for a startup company. Cannot emphasize how angry it makes me. But yeah, the potential is there. The potential for helping people with genetic disorders. There's a lot of companies out there that are doing ethical research. There's a lot of companies and researchers out there that want to take these technologies and use them for good. So it's not all gloom and doom. It can be fun. And yeah.
I was just going to say thank you so much, Maria, for being here today and for giving this talk. It was very informative, and thank you for raising all of these issues. Just to let everyone know, if you want to continue the bioethics conversation, we have a community conversations next week at 3, led by Maya Highland. So thanks everyone for coming, and thank you, Maria, for your time today.
Thank you, guys. Be curious. That's the only thing I want to leave you guys. Be curious about what's going on with these tech. Be curious what researchers are doing with them, and be part of the conversation. Just because you're not a scientist doesn't mean you shouldn't be part of the conversation about ethics and research and what's going on with gene and cell therapy.